Assalamu alaikum students. Before going to our educational topic, please subscribe my channel Muntzir Mehdi Science TV on YouTube. Press the like button and share this lecture to your friends and classmates who have no access to it to add in their science education. And those students that are preparing privately can benefit from our channel very effectively. Let's come back to our topic, which will include many terms about enzymes like complete enzymes, cofactors, coenzymes, prosthetic groups, similarly holoenzyme and apoenzyme, endoenzyme and exoenzyme. We will also study about allosteric side of an enzyme. As this video is the second part of the enzymes video lecture, so let's start with what is the complete enzyme. As we have known in our previous video lecture that an enzyme is truly made up of protein, but certain enzymes to perform their functions, to perform their jobs properly, seek some partners for themselves. For example, an enzyme requires a partner, a helper for itself. When this partner attaches with the enzyme, then the enzyme will be functionalized. Therefore, we use the term complete enzyme. This enzyme that you are watching on the mobile screens, sometimes this enzyme is not a complete enzyme. It possesses a partner with itself. When this partner associates with the non-complete enzyme, then it will become a complete enzyme. Now we understand this complete enzyme with a slight detail. So, what is a complete enzyme? It also has another name, the holoenzyme. Let's again consider this enzyme's diagram thoroughly. That is prepared by amino acid protein. So, now another partner is associated with the enzyme. Students, this new partner must be non-protein. What is enzyme? Protein. So when a protein part combines with a non-protein part, then we will get a complete enzyme. So this is our complete enzyme in which a non-protein part is completely joined with a protein part. This complete enzyme is also called the holoenzyme. So now you have completely understood the complete enzyme and holoenzymes terms. I also tell you another thing. This non-protein is a general name. It is also named another name, that is a cofactor. We will also study about cofactor in detail later. So now you can assume that the cofactor is a partner, a friend of an enzyme. When it attaches with an enzyme, then the enzyme can perform its function properly, perfectly, and thoroughly. As a functional enzyme is known as the holoenzyme. So the functional part of an enzyme, which is called the holoenzyme, this holoenzyme consists of two parts. First is the protein part and the second is the non-protein part. As I have already told you that this protein part consists of some amino acids that join together to prepare the polypeptide chain, the protein part of an enzyme, that is called the apoenzyme. Students, apoenzyme is that part of an enzyme which is synthesized by the protein part of an enzyme only. On the other hand, consider this non-protein part which is synthesized by the other groups that are called the cofactor or coenzyme. So what is this cofactor or coenzyme? It is non-protein part and is synthesized by the non-protein part of an enzyme. Again, consider from this complete enzyme, that is a holoenzyme. If we remove this non-protein part, delete this non-protein part, so that there is no non-protein part, only the protein part is remained. This protein part is named as apoenzyme. So now the enzyme is alone without any cofactor, only the protein part. But we will name this alone enzyme single enzyme. We will call it as apoenzyme. Dear students, mostly apoenzyme is a loser enzyme, disable enzyme. Therefore, it requires a cofactor, a helper to attach with it. In this way, it will become a complete enzyme, holoenzyme. I hope now you are completely understood the complete enzyme, holoenzyme and apoenzyme terms. So after the apoenzyme and holoenzyme discussion, next we will study about the cofactor. Students, when we think about enzymes cofactor, then a question emerges in our minds that what thing is the cofactor? As an enzyme is a protein part of a holoenzyme, let's imagine that it is our enzyme itself. It is made up of protein, and an enzyme to be completed require another partner that will be non-protein part, that is not made up of protein but another thing that will be non-protein. This non-protein part is the partner of an enzyme, achiever of an enzyme. This partner of an enzyme is named as cofactor. Due to this cofactor, this enzyme would become a complete enzyme. And now to this complete enzyme, we can also name as holoenzyme. The cofactors are of two types. The first ones are inorganic and the second type is organic. Inorganic cofactors mostly include metal ions such as calcium ions, magnesium ions, copper ions, and zinc ions. So these metal ions which are loosely attached with the enzymes are called cofactors, which are the partner of an enzyme. Inorganic cofactor can also include chloride ion. Although chloride ion is not a metal ion, so mostly inorganic ions, which would also be the metal ions, can be included in this category of enzymes cofactor. Either that are the positive ions or negative ions. Mostly these are the positive ions which are given here. But negative ions like chloride ions can also become the inorganic cofactors. We can also give them another name, the metallic activators, because in the old course these are known by the name of metallic activators. Dear students, due to the presence of a metal ion, a non-functioning active site can be changed into a functioning active site. So the presence of a cofactor changes the enzyme's protein shape. Its attachment allows the enzyme to create a better fit with its substrate. The cofactors of other enzymes take part in the temporary bonds formation between the enzyme and its substrate during enzyme substrate complex mechanism. So it has this brief role. Next we study the organic cofactors. Organic cofactors are of two types in the living organisms. First type is named as coenzyme and the second type is named as prosthetic groups, which means that coenzyme and prosthetic groups both are cofactors and belong to organic category of cofactors. But there is a difference in between the two. Suppose we draw a coenzyme diagram in which it is an enzyme, and here we have a cofactor that is attached to an enzyme, but this cofactor is loosely attached to its enzyme. This sign is representing that the cofactor is loosely bind with the protein part, that is an enzyme. So due to the presence of this sign, you can know that the cofactor is loosely attached to the enzyme. So when it is loosely attached, then it can release from the enzyme, detach from the enzyme surface to move to another place. In this case, the name of cofactor is coenzyme. That is a very excellent example of coenzyme is NADI, nicotinamide adenine dinucleotide. A vitamin also takes part in the NAD itself. So students, coenzymes are loosely attached non-protein part organic molecules that participates in the enzyme's catalytic activity. Coenzymes are utilized during the electrons transportation from one enzyme to another. These electrons are present in the form of hydrogen atoms. Many vitamins like niacin and riboflavin function as coenzymes, or they are utilized to prepare coenzymes. So we can say that coenzymes transport energy in the form of hydrogen atoms from one enzyme to another. One excellent coenzyme in the cell is the hydrogen acceptor nicotinamide adenine dinucleotide, which is synthesized from vitamin B. As we have known from our previous videos that NAD acquires a hydrogen atom from an enzyme and it reduces to NADH. Dear students, the electron that the hydrogen atom carries contains energy. This energy is carried by the NADH molecule. For example, when various foods are oxidized in the cell, then the cell removes electrons from the food molecules. It donates these electrons to NADH, which then reduces to NADH. Dear students, as we have discussed that many vitamins like niacin and riboflavin function as coenzymes or they are used to synthesize coenzymes. So here I want to answer a question that why vitamins are necessary for living bodies. It is said that take the vitamins because it will provide energy and power. What does mean of it? As the vitamins are essential part of our bodies, so it is a reality that all the vitamins have many different roles in our bodies. But out of these vitamins, the vitamins of vitamin B complex have remained the partner of some enzymes. And if you want that the enzymes must work in your bodies properly, then you must take the vitamins in your food properly. In this way, the vitamins must affect your body growth and development properly. Therefore, some vitamins of vitamin B complex lead to the category of coenzymes. So during the annual examination, it can be asked from you that what is the role of vitamins during the enzymes function, or what is the function of vitamins during the enzyme's job, or what is the job of vitamins during metabolism. The answer of these questions is that the vitamins act as a coenzyme, function as a coenzymes for enzymes. Next, we will discuss about the prosthetic group. That what is a prosthetic group? Students, a prosthetic group acts as a reciprocal of coenzyme or reverse of coenzyme. Let's again consider this is our enzyme. This partner will also be attached to the enzyme, which is a cofactor, which is a non-protein part of an enzyme, which is also belong to the organic category of cofactors. But the bond which is present between the enzyme and the cofactor is a strong bond that is strongly attached, which is a covalent bond. So if a cofactor is strongly attached to its enzyme, then it will belong to a prosthetic group of cofactors. Its example is FAD, flavin adenine dinucleotide. FAD is also derived from a vitamin. So students, the covalently bonded non-protein part of an enzyme, which is also an organic molecule, is known as prosthetic group, whose example is FAD, which is also derived from a vitamin. Dear students, so as we have known that NAD and FAD which are derived from vitamins, so here a question happens why some vitamins are necessary for the proper growth and development of living organisms. Its answer is that the vitamins serve as a coenzyme. Although vitamins have some other jobs, debar to act as a coenzyme that occur in laboratories, reproduction, bone marrow, and in various sites of living organisms. These act as an antioxidant, but often that vitamins which act as a coenzyme mostly are vitamin B complex. Therefore, a living organism must not provide the shortage of vitamin B complex or other vitamins to itself. You must also take the vitamins by green vegetables like spinach, fruits, cow liver, fish oil, egg white. By using all these things, you will get the vitamins which are very unique and excellent for their proper growth and development. So we can say that some enzymes require prosthetic group for their normal activities. So a prosthetic group is firmly attached to the enzyme which activates the enzyme. On the other hand, the inactive enzyme which doesn't possess a prosthetic group is known as apoenzyme disable enzyme. But the active enzyme with attached prosthetic group and coenzyme, which are the types of cofactors, organic and inorganic, is known as holoenzyme. Next, we will see the two types of enzymes, intracellular or endoenzymes and extracellular or exoenzymes. Dear students, endo means inside. So endoenzymes can act within the cell. Therefore, a large number of plant enzymes are endoenzymes which act inside the cell, whereas exoenzymes diffuse out of the cell through the plasma membrane and function on some outside medium. Exoenzymes act on the substrates like fungi, bacteria, and in some of the insectivorous plants. Dear students, exoenzymes are synthesized by the living protoplasm, but they do not require the environment of a living cell for their action, for their function. Next, we will see what is an allosteric site. Students, allosteric site is present on the surface of some enzymes, and there are some specific effectors which can bind on this allosteric site. An enzyme which possesses this allosteric site is known as allosteric enzyme. An allosteric enzyme can change its structure when a specific effector comes to bind with it. It is called allosteric modulation. Students, this modulation can be direct or indirect. Direct or indirect modulation means the effector may bind directly to the binding site of an enzyme, or the effector may bind to other proteins or protein subunits, and then this protein interacts with the allosteric enzyme and thus increases the catalytic activity of a chemical reaction by the allosteric enzyme. So students, it is second part of enzymes video, during which we have understood different terms of enzymes, endoenzymes and exoenzymes, and also we have understood the allosteric site of an enzyme. Dear students, cofactors, coenzymes, prosthetic group, apoenzyme and holoenzyme are often important topics that come during annual examination. These questions come in the form of short question, so prepare it with excellent consideration. At the end, I want to encourage you that concentrate your precious education, that is the future of not only yourself but also your country, your nation, your civilization, and also your religion. So make that only leader which can change the future of a nation. Lastly, many thanks for watching this your video.